My name is Angelina Cicerchia, and I'm a senior at UMass Amherst, doing a senior thesis in the lab of Dr. Mandy Muller. The title of my project is The Role of HuR in RNA Stability during Kaposi Sarcoma-Associated Herpesvirus infection. Kaposi Sarcoma-Associated Herpesvirus, also known as KSHV, is the causative agent of Kaposi sarcoma, along with other B-cell lymphomas. Kaposi sarcoma is the most common cancer in individuals living with HIV/AIDS today. Kaposi sarcoma is a cancer that causes lesions in soft tissue, such as tissues of the skin, mouth, and internal organs. KSHV lives in a biphasic lifestyle with a latent and lytic phase. Upon KSHV infection in a host, the virus is in the latent phase, where its goal is to maintain being hidden from the immune system. This is how the virus can trigger lifelong infection. But from time to time, the host can become immunocompromised, causing KSHV to sense stress. This triggers activation of the lytic phase, in which there is amplification of the viral genome. The virus's goal is to create progeny and exit the host. This needs to happen quickly, so one mechanism that KSHV utilizes is host shutoff. Host shutoff involves the widespread degradation of host RNA with the purpose of freeing up host cell machinery for viral usage. KSHV drives host shutoff through the expression of the endonuclease SOX. Endonucleases are proteins that can cleave nucleotides. SOX can degrade about 80% of host RNA transcripts, but this leaves about 20% of transcripts that can escape SOX. Our lab is interested in the 20% of transcripts we call escapees. Previous research located a component on the 3' untranslated region of escapees we call the SRE, which stands for SOX-resistant element. This allows transcripts to actively escape SOX. This leads to the question of, how do these SRE-containing transcripts escape SOX degradation? The SRE was found to be bound to an array of cellular proteins that are likely to contribute to mRNA stability. My project focuses on one of these proteins, which is human antigen R, also known as HuR. This protein is known to promote RNA stability when bound. HuR is an mRNA binding protein that is known to bind to regions on RNA transcripts that are AU-rich elements. On the left is a depiction of the organization of HuR and its domains. So HuR is made up of 3 RNA recognition motifs known as RRM domains. RRM1 and RRM2 are divided by a linker of 12 amino acids, and RRM2 and RRM3 are divided by a hinge unit that contains a nucleoplasmic shuttling sequence. On the right is a depiction of a general movement of HuR during a pathogen detection in a cell. A pathogen is usually detected by the innate immune cells, and transcription of interferon-stimulated genes occurs. Pathogen detection can also cause HuR to be phosphorylated on the hinge region, which contains that nucleoplasmic shuttling sequence. And this causes HuR to move from the nucleus to the cytoplasm. Once in the cytoplasm, HuR can bind to RNA at an ARE region on the 3' UTR, and this binding promotes RNA stability and increases the occurrence of translation for these transcripts. Knowing the general purposes of HuR in cells allows me to address some aims of my project. My first aim is to determine how SRE protection is mediated by this HuR protein. Different mechanisms could be along the lines of HuR offering physical protection from SOX, or even HuR binding and shuttling these SRE-containing transcripts to a location or pathway inaccessible to SOX. My other aim is to figure out which parts of HuR bind to the SRE and lead to protection from SOX degradation. As I explained before, HuR has different binding domains. We are interested in locating the specific domains that are necessary to preserve the protective phenotype. This leads me to further analyze HuR domains with the establishment of mutants. The creation of these mutants will allow us to conduct experiments to determine the region necessary for SRE protection. As I described before, HuR is made up of 3 RRM domains. The first mutant represents the full-length HuR. Each mutant has different RRM domains deleted, and all the mutants are flag tagged, which will help us to track the mutants during experiments. The right image is a Western blot, and Western blotting is a way to look at protein levels in cells represented by little bands. Cells were transfected with each mutant and then harvested to extract the proteins. Staining with anti-flag antibodies allows us to see the mutants expressed at correct sizes in our cells. And now that I have these mutants established, I can move on to the next step of knocking down HuR, which will allow me to analyze effects of RNA degradation in relation to HuR expression. I perform knockdown using siRNA, which is double-stranded RNA that binds to complementary RNA in cells and leads to silencing. After siRNA transfection in cells, I visualize samples with Western blot analysis. Looking at the Western blot on the left stained with anti-HuR antibody, we can see that HuR was effectively knocked down based on the faint to non-existing bands in the samples treated with the siRNA. The most right 2 samples have the addition of either full-length HuR or the delta RRM3 mutant, in addition to being treated with the siRNA. The dark bands in either of these 2 lanes represent the HuR mutants that were added to the cells, and not the endogenous HuR that was knocked down with siRNA. The Western blot on the right has the same samples stained with anti-flag antibody. This allows me to confirm that the bands in the other blot represent the mutants, since they are flag tagged. Overall, I know that siRNA works to knock down the HuR, and that my mutants are resistant to the siRNA silencing. My current project conclusions are that HuR is an important element in the escape of SRE-containing transcripts from SOX degradation. I've been able to show that our HuR mutants express in cells and that HuR can be knocked down with siRNA. Our mutants are also resistant to siRNA silencing, which allows me to use them during siRNA silencing experiments and address my project aims. I know that HuR is needed for SRE protection, so my goal is to determine what happens when HuR is eliminated and rescued with our HuR mutants. My future directions include using qPCR to measure RNA levels after the transfection of SOX and HuR mutants in cells treated with siRNA to knock down HuR. I expect to see a decrease in RNA levels in samples that have an important region of HuR deleted, since HuR will no longer be able to perform its protective function. Another future direction is to use nuclear fractionation to track movement of HuR during the course of KSHB infection. Immunofluorescent microscopy will also allow me to visualize the movement of HuR in the future. Overall, research into host shutoff mechanisms of KSHB will allow for the development of therapeutics against cancers caused by KSHB, and even other gamma herpes viruses in general. I'd like to thank all of the Muller Lab, especially Dr. Muller and Daniel, for always answering my questions and helping me grow as a researcher. Thank you all for listening to my presentation.